The California Mastitis Test, or CMT, is a rapid animal side test to detect subclinical mastitis. Mastitis is an inflammation of the mammary gland, usually produced by microbes. With inflammation, the mammary gland capillaries increase in permeability, and constituents of the blood leak out from the capillaries and enter the gland and the milk. The CMT works by detecting increased somatic or body cells in milk. With the test, the cells being measured are mainly neutrophils. Neutrophils are present in increased numbers in the udder when the animal has mastitis. The CMT is most often used to detect mastitis in the individual quarters of the cow in order to detect subclinical mastitis. It can also be used to conduct tests on bucket and bulk tank milk samples to estimate the somatic cell count of the entire herd. For the individual cow or goat, the CMT is used as part of a complete physical examination to test for the presence of subclinical mastitis. It is not necessary to use the CMT when clinical mastitis is present, as you already know that the test will be positive. For use in a herd, the CMT can aid in screening cows to determine which cows may be infected or contributing to a high cell count. You can estimate the approximate cell count from the degree of CMT reaction. The degree of reaction, or CMT score, can also be used to estimate the percentage of milk loss or percentage of cows infected. Tables are readily available which help estimate these measures. To conduct the test, you will need a 4-cup CMT paddle and CMT reagent. The CMT reagent contains a detergent to lyse the cells, releasing DNA, and a pH indicator, bromocresol purple. DNA from lysed cells produces a change in the consistency if the cell count in the sample is elevated. The pH indicator detects a change in pH. The test detects changes associated with inflammation, including increased inflammatory cells and changes in milk due to inflammation. One of those changes is an increase in pH, detected when the pH indicator changes to a more purple color. Begin by cleaning the major debris from each teat. It is not necessary to prepare the quarter as you do for a septic collection of a milk sample. First, 2 streams of milk are expressed from each quarter. The very first milk is the highest in somatic cells, so we want to remove that milk. Within each cup are large and small circles, which are used for measuring. Next, squirt approximately 2 milliliters or 1/2 teaspoon of milk from each teat into each appropriate paddle cup. By convention, the handle of the paddle is pointed towards the head of the cow. Tilt the paddle nearly vertical, allowing excess milk to spill out until the milk is level with the outside of the largest circle in each cup. Next, tilt the paddle down until the milk is halfway between the 2 circles. Then add equal amounts of CMT reagent into each cup until the combined level reaches the inner circle. Again, this will be 2 milliliters or approximately 1/2 teaspoon. Hold the paddle level and rotate it in a circular motion for no more than 10 seconds to thoroughly mix the contents. The CMT reagent reacts with the lysed neutrophils, and the mixture thickens or gels in response to the amount of cells present. A color change can also occur. The reaction should be read within 20 seconds as the visible reaction disintegrates after this amount of time. Reactions are scored on 5 levels. Negative shows no thickening and is homogeneous. Trace shows slight thickening that may disappear with swirling. Level 1 presents distinct thickening but no gel formation. Level 2 thickens immediately, begins to gel, but levels in the bottom of the cup. With Level 3, a distinct gel is formed, and surface elevates with a central peak above the mass. An alternative scoring system is to use categories of negative, trace, positive, and strongly positive. Mastitis control programs are intended to reduce mastitis levels in a herd and to prevent new intramammary infections from occurring. To help prevent mastitis, ensure that cows and their environment are kept clean. Regularly clean and maintain milking equipment. Good milking practices are especially important. Teat dipping cows after milking is essential. Appropriately practiced mastitis control programs have been shown to reduce mastitis in a cost-effective manner.